Eating food provides fuel to our body. Through the process of digestion, the food will be broken down into carbohydrates and other molecules. These nutrients will be transported through blood circulatory system. From there, they will enter a tissue cell to undergo the process of cellular respiration. Cellular respiration is the process of taking the food we eat, then converting it into the energy that can be used by the cell. It will turn the glucose into carbon dioxide, water and high energy compounds which are ATP and NADH. Cellular respiration starts with the first process called glycolysis. Glycolysis means splitting of one molecule of glucose into two molecules of pyruvates through a series of reactions catalyzed by specific enzymes. Glycolysis itself consists of two words. The word glyco means glucose and the word lysis means breakdown. The glycolysis is a metabolic pathway that involves systematic breakdown of glucose to produce energy, and glycolysis takes place in the cytosol of the cell. First, we will have an overview about this process, and then we'll study about the various reactions involved. In glycolysis, one molecule of glucose, which is a 6-carbon compound, is broken down into two molecules of pyruvates, which are 3-carbon compounds. During this conversion, energy is released in the form of ATP and NADH. The pyruvate form is further metabolized in various energy pathways that produce more energy. The process of glycolysis takes place in two phases. In the first phase, the glucose is converted into intermediate compound. Since this conversion involves addition of phosphate, therefore energy in the form of ATP is used. In the second phase, the intermediate will be converted into final product, which is pyruvates. During this phase, energy is produced. Now let's have a look at the various reactions involved in the glycolysis. So it all begins with a molecule of glucose that enters the cytosol. The first reaction that takes place is phosphorylation, in which glucose is converted into glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. Hexokinase transfers a phosphate group from ATP to the glucose. In this reaction, ATP is converted into ADP, hence it utilizes one molecule of ATP. This phosphorylation makes glucose more chemically reactive. Next, the glucose 6-phosphate is converted into its isomer, the fructose 6-phosphate. This is the isomerization process, a simple rearrangement reaction. In the next step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate through phosphorylation process. That is the addition of another phosphate group at the first carbon of the fructose ring. This reaction is catalyzed by the enzyme phosphofructokinase. Again, ATP is converted into ADP. Hence, another ATP is utilized in this reaction. Next, the fructose 1,6-bisphosphate is split into two molecules, the glyceraldehyde 3-phosphate, G3P, and the hydroxyacetone phosphate, DHAP. These two compounds are 3-carbon sugars. Both the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are actually isomers and can be interconverted to each other by isomerization process. But since the glyceraldehyde 3-phosphate is further utilized in the next process of glycolysis, dihydroxyacetone phosphate is enzymatically converted to its isomer, glyceraldehyde 3-phosphate. These 5 steps compose the first phase of glycolysis, which is the energy investment phase, where the energy is consumed in this process in the form of 2 ATPs. Keep in mind, during the first phase of glycolysis, the energy investment phase, every molecule of glucose is split into 2 molecules. Therefore, every reaction in the second phase happens twice. Thus, the number of products will be double. In the next reaction, each molecule of glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. The processes involved are oxidation and phosphorylation. During oxidation of glyceraldehyde 3-phosphate, the coenzyme NAD+ is reduced into NADH plus H+. Next, the energy released from this exergonic redox reaction will be used for phosphorylation, where a molecule of inorganic phosphate present in the cytosol will be attached, forming 1,3-bisphosphoglycerate. After that, the 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. A phosphate group will be transferred from each 1,3-bisphosphoglycerate to ADP to generate ATP. This production of ATP happens through substrate-level phosphorylation. Next, the 3-phosphoglycerate is rearranged into 2-phosphoglycerate through isomerization process. Then, the 2-phosphoglycerate is converted into phosphoenolpyruvate. In this step, a water molecule is removed from each 2-phosphoglycerate, forming double bond. In the final step of glycolysis, the phosphoenolpyruvate is converted into pyruvate. A phosphate group in the phosphoenolpyruvate is transferred into ADP and converted into ATP. This production of ATP happened again through substrate-level phosphorylation. These all 5 steps compose the second phase of glycolysis, the energy payoff phase. As you can see, energy in the form of ATP is produced in this second phase. Now, let's summarize the process of glycolysis. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes. The net reaction of glycolysis is summarized in this equation. We metabolize a molecule of 6-carbon glucose that splits into 2 molecules of 3-carbon compound, pyruvates. There's net utilization of only 2 ADPs. Besides, 2 molecules of inorganic phosphates are utilized in this step. 4 ATPs are produced in the second phase. The first 2 ATPs during conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate. Then, the remaining 2 ATPs are generated during conversion of phosphoenolpyruvate into pyruvate. Meanwhile, during the first phase, 2 ATPs were utilized in 2 steps. Firstly, during conversion of glucose into glucose 6-phosphate, and secondly, when fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Therefore, there is a net production of 2 ATPs at the end of glycolysis. Then 2 molecules of NAD+ are reduced into 2 molecules of high-energy electron-carrying molecules, named NADH plus H+. This occurs when glyceraldehyde 3-phosphate is oxidized and phosphorylated into 1,3-bisphosphoglycerate. When oxygen is present, the pyruvate and NADH plus H+ will enter the mitochondria and the next stage of cellular respiration begins. This is crucial to produce more energy to fuel our living processes.